Hi, Miss K. This is our Eco Column Screencast. I'm Claire.
I'm Gabby.
And I'm Lina.
Okay, so we're going to look at our timeline for our Eco Column.
So on day one, the aquatic chamber had 2 live fish, 1 orange one and 1 with a white underside, and 2 Elodia plants. The decomp chamber, the bottle looked humid, but there was no clear decomposition yet. And the terrestrial chamber was just dirt with no visible plant growth. And here are pictures. Here's the aquatic chamber. Here is the decomposition chamber. And here is the terrestrial chamber. And here's the entire eco column all together. So day 2, the aquatic chamber, both fish were still alive and they were eating at the plant. There was condensation at the top of the chamber. In the decomposition chamber, it was humid looking, there was condensation, but there was no visible decomposition yet either. And in the terrestrial chamber, there were no visible changes. So here's our aquatic chamber. The water's looking clear still. The decomposition chamber looks humid. And the terrestrial chamber has no stems growing yet. And this is the entire column. Day 3, one of the fish died in the aquatic chamber, and it was the one with the white belly, and its body was tangled in the plant. The water was clear, and the plant was a vivid green. In the decomposition chamber, there was black mold on the wall. Decomposition had started on the apple, and it was humid. And in the terrestrial chamber, there were small sprouts growing. So here is our aquatic chamber, and the decomposition chamber, and the terrestrial chamber, and the entire chamber together.
Okay, so on day 4, both fish were dead in the aquatic chamber, which is another reason why there was high nitrogen levels, but the Elodia plant was still alive and the water was clear. In the decomposition chamber, more mold was appearing, organic matter was breaking down. And in the terrestrial chamber, there was 8 visible sprouts up to 2.5 inches tall. So here is a picture of the aquatic chamber. The fish is dead and sad, but it's at the top of the chamber. This is the decomposition chamber. This is the terrestrial chamber, and this is the whole eco column as a whole. Okay, so on day 5, the aquatic chamber, the 2 fish were still dead. The plants were bare at the bottom of the stems. In the decomposition chamber, there was lots of mold, both black and white, and the bottle still looked very humid. In the terrestrial chamber, there was 9 sprouts visible, some bigger than others, and the tallest one was 3 inches tall. So here's the aquatic chamber, the decomposition chamber, the terrestrial chamber with the sprouts, and the whole eco column as a whole.
On day 6, in the aquatic chamber, the fish were decomposing and the eyes were turning black. The bodies were not moving. In the decomposition chamber, there was a lot of mold, and in the terrestrial chamber, 10 sprouts were visible and the plants were thriving. This is the aquatic chamber. This is the decomposition chamber. This is the terrestrial chamber, and this is it as a whole. On day 7, in the aquatic chamber, the water appeared murkier and was turning slightly green-brown. In the decomposition chamber, the leaf mold was significant, and in the terrestrial chamber, the plants were very green. So this is the aquatic chamber, the decomposition chamber, the terrestrial chamber, and all the chambers together.
So now we're going to look at the graphs. So here are the temperature changes for the aquatic and terrestrial chambers. The blue is for the aquatic and the red is for the terrestrial. As you can see, as the days increased, the terrestrial chamber generally decreased in temperature, starting somewhere around 22.5 and ending at around 21. The aquatic chamber, however, had a slight dip and then started increasing again as the days went on. It started at almost a 22, dipped on day 4 closer to a 21, and on day 7 went back up to a 21.5. For the pH in the aquatic and terrestrial chambers, the red, which signifies the terrestrial chamber, it stayed pretty even at about 6.8 to 7 in that general vicinity. For the aquatic chamber, the pH started at a 5.6 and then increased up to a 6.2 by the end of day 7. And then for the changes in nitrogen, phosphorus, and turbidity in the aquatic chamber, for turbidity, which is shown by the orange, we were unable to get a numerical value on the first day due to time limit, so we kept the line at 10, which is what we got on the last day. For nitrogen, it started off at a 2 and decreased to a 1 by the end, and phosphorus started out at a 1 or at 0.8 and went up to a 1 by the end. And then changes in nitrogen, phosphorus, and potassium in the terrestrial chamber. Nitrogen decreased as the days went on. Phosphorus increased, and potassium slightly decreased.
Okay, so these are some data tables for the aquatic chamber. So basically for the temperature, the average was around 22.4. The maximum temperature we found was 22.9 and the minimum was 21. So there wasn't that much of a huge gap between them. The pH average was about 5.87. The maximum was about 6.19 and the minimum was 5.53. The salinity was, the average was 59.8. The maximum was 80. And the minimum was 53, so there actually was a huge change in that one. For the dissolved oxygen, the average was about 2.57, the maximum was 4.4, and the minimum was 1.1. Again, there was a huge difference between the two of those. Okay, so for the terrestrial chamber, the temperature average was 21.56, the maximum was 22.6, the minimum was 21.1. The pH average was 6.9, the maximum was 7, and the minimum was 6.8. So as you could tell from the graph before, there wasn't that much of a change between the pH throughout the days of the terrestrial chamber. Okay, for the aquatic chamber, again, the average was, for the temperature was 22.4. The maximum was 22.9, the min was 21. And the pH average was 5.87, the max was 6.19, and the min was 5.53. Okay, so for the terrestrial chamber, the nitrogen average was 50, the maximum was 60, and the minimum was 40. For the phosphorus, the average was 175, the maximum was 200, and the minimum was 150. The potassium average was 182.5, the maximum was 185, and the minimum was 180. So for the potassium, there wasn't that much of a change, but we could definitely see more of a change between the phosphorus because there was a bigger gap. Okay, so for comparing our ecosystem to a real ecosystem, well, obviously our ecosystem was indoors, so we were limited by a lack of sunlight. We were watering it frequently, so it wouldn't really be accurate in like outside in real life because sometimes there's going to be more rain than others, and there was a lack of decomposers. We didn't have any actual living decomposers in our decomposition chamber, and there was only a couple of fish, only 2. And in a real ecosystem, there would be lots of sunlight depending on where you are. There'd be more precipitation, there would be actual decomposers, and there'd be hundreds of different organisms making up a community rather than just a couple. Okay, so some of the limiting factors. For the terrestrial chamber, there was the lack of rain. We were indoors, so there wasn't that much sunlight. The temperature was based on the room temperature rather than what would actually be based on the biome, wherever it was located. In the aquatic chamber, some of the limiting factors would be the salinity, the nutrients of the plants, the lodia, and the temperature of the water, which was room temperature. And in the decomposition chamber, some of the limiting factors were the precipitation, and there was no insects or bacteria placed which could help break down the food scraps. So basically it was just the mold in ours rather than having like worms or any kind of organisms that we could have been there.
For succession, it showed in the form of mold in the decomposition chamber. Bacteria in decomposition column may have raised the pH and temperature of the column, which allowed mold to flourish in the new environment. So that was evidence of succession. The competitive exclusion principle, we did not see any evidence of it in our eco column because we did not witness two different species competing over a resource. The goldfish were of the same species and they had plenty of resources for both.
Okay, so ecosystems in the classrooms. So other ecosystems had different organisms. Some had snails, some had bigger or smaller fish. Some of the organisms lasted longer than other. Both our fish were dead after 9 days, while some other groups lasted the full 2 weeks, or one would die after just 1 day. Some had different types of soil. Some were nutrient rich than others. And some of the ecosystems got more sunlight based on where it was placed in the classroom.
As for the law of tolerance, did we observe signs of law of tolerance in action in our eco column? We did. The goldfish died because they experienced a factor that was too high or low for their range of tolerance, whether it be temperature or food or the nutrients in the water. The plants in the the plants in the terrestrial chamber survived because the factors within the eco column were in their range of tolerance, so whatever was happening in the columns below them was working in their favor and allowed them to thrive. And then mold grew in the decomposition chamber because the environment was suitable for them. The temperature was okay, and they were able to grow onto the items in there because it was in their favor.
So the roles in our ecosystem include that the decomposers were decomposing the fish after they died, and the mold that grew on the organic matter in the decomposition chamber. The producers included the sprouts that grew up in the terrestrial chamber and the elodia in the aquatic chamber, and the consumers were the fish when they were alive. They all played an important role in the food web, and one could not have been able to survive without the other. So on the left here, you can see is the nitrogen cycle. And so at the top, nitrogen fixation occurred when the bacteria in the soil converted nitrogen gas into ammonia and ammonium, and then more bacteria converted this into nitrate ions, and then assimilation occurred as these nutrients were taken up by the plants. And in ammonification, which occurred in the decomposition chamber, the decomposers converted the nitrogen-containing compounds into ammonia and ammonium, and then in the aquatic chamber, denitrification occurred, which only happens in waterlogged areas, and this is where they converted ammonia and ammonium into gaseous nitrogen, and then here nitrogen fixation could occur once again with bacteria. And throughout the entire eco column, the nutrients flowed down as we poured water down, so that's how nitrogen was able to get to the bottom. And on the right we have the carbon cycle, and so CO2 was absorbed through the plants and stored there through photosynthesis. And then CO2 was also released from the organic matter as it was decomposed. And in the aquatic chamber, there was some CO2 that was stored in the water, and the fish released CO2 through aerobic respiration, and the plants absorbed it through photosynthesis. And the food chain includes that the bacteria ate and decomposed organic material, and including the dead fish, and that the fish ate the plants in the aquatic chamber.
Thank you.